The Zoonomia Consortium. Thanks, Allison. Sorry about that. The Zoonomia Consortium has sequenced and aligned 240 genomes from 240 different mammals. And this then provides us with an incredible opportunity to try to understand the mechanisms underlying some of our phenotypic diversity by comparing genomes from different mammals. To motivate how we do this, I'm going to talk about the phenotype of brain size relative to body size. So really large brains relative to body size have evolved multiple times throughout mammalian history. Specifically, if we plot the body weight on the x-axis and the brain weight on the y-axis, we can see that there's generally a linear relationship between body weight and brain weight. Yet, some animals, like humans and dolphins, have much larger brains than we would expect based on our body sizes. While the blue whale, which is actually, dolphins are actually much more closely related to blue whale than they are to humans, has a small brain relative to its enormous body. Specifically, we quantify the deviation from this linear relationship of body size versus brain weight as the brain size residual. So the question we want to ask is, how have these large brain size residuals evolved in multiple points of mammalian phylogenetic history? Fortunately, to aid our investigation, in addition to many genomes from Zoonomia, a massive effort was done a few years ago to quantify brain size residual in a large number of mammals. Specifically, they annotated the brain size residual in 1,552 species. This includes 28 extant taxonomic orders and 150 of the Boreoeutherians. The Boreoeutherians are all the species except for the top 5 orders in this diagram in the Zoonomia multi-species alignment. And here you can see for the largest orders, they studied the variation in brain size residual. And what you may notice is cetaceans, which are the whales, have the largest variation. So then the question is, how can we use this amazing effort of curating all these brain size residual annotations along with the Zoonomia genome sequences to study the variation in brain size residual and try to figure out how this has happened? And the first approach that we considered is to look at variation in protein coding sequence. So is there variation actually within genes that code for proteins that seems to be associated with the evolution of brain size residual? So my lab mate Morgan Werflin set out to do this, and unfortunately, she was not able to find any protein coding regions associated with the evolution of brain size residual. And what this suggests is that brain size residual has evolved at least in part, and maybe entirely, through changes in gene expression. So to explain what I mean by this, let's imagine an enhancer. So we've discussed in other talks that enhancers are regions of the genome, usually outside of genes, that loop. We've been talking a lot about 3D interactions, 2 transcription start sites to help recruit RNA polymerase to then activate gene expression in a specific tissue or cell type. And these enhancers are bound by proteins that were mentioned in the last talk called transcription factors. And because of transcription factors protein sequence, they recognize very specific combinations of A's, T's, G's, and C's. So let's imagine that we have in humans, which have a very large brain size residual, this enhancer in the brain activating gene expression in the brain in a gene that controls brain size. Now let's imagine our closest relative, the chimpanzee, which has a smaller brain size residual. Now in the chimpanzee, there might be a sequence difference in this enhancer. And because transcription factors recognize specific combinations of A's, T's, G's, and C's due to their protein structure, this could then lead to a difference in transcription factor binding and the enhancer, which could lead the enhancer to no longer be able to recruit RNA polymerase, which could then lead to a decrease in gene expression of the gene it regulates in the brain, and thus a decrease in brain size. This therefore shows that there are actually 2 ways we should be using genome sequence to study phenotypic diversity. First, as I mentioned before, by comparing protein coding sequences, but second, we should also be comparing enhancer sequences. A previous study set out to find non-protein coding regions, potential enhancers associated with brain size evolution. And they did this by looking at human-specific deletions, so regions of the genome that are lost in humans but present in other mammals. And they found a specific example here. And then they looked at the chimp ortholog and found, there was some mention of luciferase assays in early talks, that this region in chimpanzee is able to activate gene expression in the brain. Now, this region in chimpanzee is near a tumor suppressor gene. So their hypothesis is that in chimpanzee, this tumor suppressor gene gets activated and then prevents the brain from growing to be as large as the human brain, which does not have this enhancer. So we wanted to ask the question, is this enhancer also lost in our bottlenose dolphin and a harbor porpoise, which are the two cetaceans with very large brain size residual? So we used the new genomes from Zoonomia. These cetacean genomes were not present in the previous study to try to investigate this. And to our surprise, this region is actually present in all of these cetaceans in Zoonomia, which suggests that this in human-specific deletion is not the entire brain size evolution story. What this means is that to really understand how a phenotype has evolved, we need to be able to get an enhancer activity in a relevant tissue in a diverse set of species. So a previous study has set out to use data from large numbers of species to identify regions of the genome associated with the evolution of phenotype. This method is called RAR converge. It can be applied to both protein and non-protein coding sequences. And the idea of RAR converge is to find regions of the genome with a high relative evolutionary rate in species with a phenotype relative to species without a phenotype of interest. So the previous study identified a set of non-coding regions with a high relative evolutionary rate in mammals that live underground. So here you can see on the x-axis, we have a relative evolutionary rate. Each row is a species, and our red mammals are subterranean mammals, and you can see that this particular region has a high relative evolutionary rate in our subterranean mammals. So they then looked at what this region of the genome might be doing, and specifically they looked at the mouse ortholog, where mice do not live underground, and they found that the mouse orthologs of these regions overlap eye-specific enhancers. So what this suggests is that these regions that seem to be faster evolving in subterranean mammals are enhancers in the eye, and these eye enhancers are maybe becoming less important when mammals live underground, which makes sense because mammals live underground, don't need as good vision. So we said, okay, let's use RAR converge to identify non-coding regions associated with the evolution of brain size. Unfortunately, we were unable to find anything. And this was a really disappointing result, but it points to a larger issue, which is that RAR converge uses nucleotide level sequence conservation, and a lot of other methods for identifying enhancers associated with the evolution of phenotypes are also using nucleotide level sequence conservation. But nucleotide level sequence conservation is actually not equivalent to enhancer activity conservation. To show you some examples of this issue, I want us to consider two regions of the mouse genome. So our lab used an assay called a TACIT. In previous talks, we've heard about DNA hypersensitivity, which assay is basically the same thing. This assays whether regions of the genome are accessible, so not wrapped around nucleosides, to be bound by transcription factors and thus regulate genes. So we can think of a region with strong ATAC-seq signal as a candidate enhancer. So here we profiled ATAC-seq in two different brain regions, the motor cortex and the striatum. You can see that both of these regions in the mouse have strong ATAC-seq signals. When we look at nucleotide level conservation scores, you can see that this first region generally has low conservation scores. So this nucleotide level conservation scoring method is called FastCons. It outputs a probability that a region is conserved, so it's a number between 0 and 1. You can see that in almost every one of these 500 base pairs, the FastCon score is much closer to 0 than it is to 1. In contrast, in this region, you can see that most positions have very high FastCon scores. So now I want you all to do an exercise with me. Let's now imagine the macaque ortholog of each of these regions. So the corresponding region in macaque. do we think for this first region with strong ataxic signal, but very low FASTCon scores, do we think that the macaque ortholog is going to be a brain enhancer? Raise your hand if you think the macaque ortholog of this first region is going to be a brain enhancer. No takers. Raise your hand if one. Okay, raise your hand if you think this macaque ortholog, very low conservation sequence, nucleotide level sequence conservation here, is not going to be a brain enhancer. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Awesome. Okay. Now let's consider the second region. We have, once again, strong brain ataxic signal, and here we have very high nucleotide level conservation. So raise your hand if you think the macaque ortholog is going to be a brain enhancer. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Raise your hand if you think the macaque ortholog is not going to be a brain enhancer. 1. Okay. So the very surprising result is that this first enhancer with super low nucleotide level sequence conservation, the macaque ortholog is a brain enhancer. But the second region, which has very high nucleotide level conservation, the macaque ortholog is not a brain enhancer. And what this shows is that nucleotide level conservation scores are not sufficient for evaluating enhancer activity conservation. So why is this surprising result happening? There are a few potential reasons for it. First is that most mammalian enhancers have tissue-specific activity, but conservation information is tissue agnostic. So if you think about that second enhancer on the previous slide, it is actually near a gene that is expressed in many different tissues. So it's totally possible that with these high conservation scores, that enhancer's activity, the macaque ortholog, is actually an enhancer in many different tissues. It's just not an enhancer in the brain, and the brain is what we're interested in for understanding the evolution of brain cells. The second reason is that enhancer function can be conserved even when nucleotide level conservation is low. And this gets at that first example. We had low nucleotide level conservation, but enhancer activity conservation. So a previous study found a specific example of this. This is an enhancer whose activity is conserved for mammals all the way out to sponges. And what's happening here is that each of these little boxes is a transcription factor binding site. So what you see is that all of the same transcription factor binding sites are present in human, zebrafish, and sponge, enabling the enhancer activity to be conserved. However, the order of these transcription factor binding sites is different in these different species. So if you go base by base, you're going to see that most bases are not actually conserved. And that might explain what's going on in that first example on the previous slide. So what we can think of these transcription factor binding sites as sequence features. So these are specific combinations of A's, C's, G's, and C's that bind transcription factor. They're features within our sequence that are related to enhancer activity. And what this result suggests is that sequence features, where by sequence features we mean transcription factor binding sites, the conservation of sequence features, though not necessarily the order, is actually indicative of tissue-specific enhancer activity conservation. Therefore, how should we be identifying enhancers associated with phenotypes involved in gene expression? One option, as I mentioned, is nucleotide level conservation. And while this has worked well for some phenotypes, for our purposes and for many others, as I've shown you, it's not going to be very useful. Another option is to experimentally assay enhancers in tissues involved in a phenotype. So we can actually experimentally use assays like a TACIT to evaluate the enhancer activity in many different species. But the reality is that we cannot obtain most tissues from most mammals, and as I mentioned, many enhancers are tissue specific. So this is usually also not a good option. We therefore needed to develop a new framework to efficiently trace the evolution of phenotypes that are evolved through gene expression. And we call this framework TACIT. I'm going to begin by telling you about the general methodology underlying TACIT. I will then tell you about how we evaluate machine learning models for TACIT. After that, I'll tell you about how we apply TACIT to brain size. And finally, I'll tell you about how we apply TACIT to some other phenotypes. So let me start by telling you about the TACIT methodology. The idea of TACIT is what we can obtain is a matrix that looks like this top one. So you can see here that each row is an enhancer, and each column is a species. and we can get enhancer activity data for only a small number of species. But these enhancer activity data does give us information because we can look at the sequences underlying these enhancers to get a sense of what sequence patterns might underlie enhancer activity. And then what we can do is we can use a machine learning model to then learn what these sequence patterns are and then predict enhancer activity in a large number of species, enabling us to fill in a lot of the missing data in this matrix. And the reason we can do that is that machine learning models can learn important sequence patterns for enhancer activity. So here we have a toy example of a brain enhancer. And what we would expect is that the machine learning model would learn that the nucleotides in large letters here are important for brain enhancer activity because these are the nucleotides that the transcription factors FOS and MEF2C bind, and these are two very important transcription factors in the brain. So the way TACIT works is that we first identify, for brain size, we first identify brain enhancers in some species. We then use the sequences underlying those enhancers to train a machine learning model. We then use our machine learning model to predict brain enhancer activity at orthologs across species. We then associate our predictions with a phenotype of interest, and then we compute a p-value to quantify the statistical significance of our association. And this framework is called the Tissue-Aware Conservation Inference Toolkit, which we abbreviate as TACIT. So I'll now go through each of these steps of TACIT in more detail so you can understand exactly how we're doing it and what drives the decisions we made for how we implement each step. So first I'll tell you about this first step of TACIT. My lab mates Morgan Worthland, Alyssa Lawler, and Ashley Brown obtained open chromatin data through a lot of hard experimental work in 4 to 5 species from each of 2 tissues, the motor cortex and liver. And for the liver, we supplemented this with some publicly available data. I'm going to tell you, for the purposes of this talk, about our results for motor cortex, because that is the tissue most related to brain size. And I'd be happy to tell you about our liver results offline. So now that I've told you about the data we generated, I'm going to tell you more about our sequence-based machine learning models, which is the second step of TACIT. So to explain a little more how this works, we talked about machine learning a little bit more before in the conference, but I'm just going to tell you how this works for DNA sequence. So what we have is we have DNA sequences that are enhancers. So in this toy example, you can see these are marked by pluses, and we call these our positive set. We also have DNA sequences that are not enhancers in our tissue of interest. We call these our negative set. And what we want to do is figure out what separates, what differentiates between our positive set and our negative set. So the first question we ask is, what kind of machine learning model do we want to use? And we've talked about a few different machine learning models at this meeting. And the kind of machine learning model we chose to use was actually mentioned by Kathleen yesterday. It's called a deep convolutional neural network. And what deep convolutional networks do is they take raw DNA sequence, so just a combination of A's, T's, G's, and C's, and they output a probability, so a number in the range 0 to 1, that the sequence is an enhancer in your tissue of interest. And we later on use these probabilities as continuous values when associating enhancer activity with phenotypes. So how do convolutional neural networks work? Here you can see an outline of a toy example of a convolutional neural network. And now I'm going to walk you step by step through each part of this so you can understand exactly what this convolutional neural networks for predicting enhancer activity are doing. So the very first thing we do is we take our sequence of A's, T's, G's, and C's, and we can create a one-hot encoding. Sorry, this projector screen's cutting it off a little, but basically what this is a matrix. It is 4 high, and the width is the width of the sequence. So in our first row, what we have is a 1 in every position where our sequence is an A, and a 0 in every other position. In our second row, we have a 1 in every position where our sequence is a C, and a 0 in every other position, and so on. So once we have this one-hot encoding, we then have what's called a convolutional layer. And for those of you who are familiar with transcription factor binding literature, you can think of a convolutional layer as a series of transcription factor motif detectors. So the convolutional layer consists of multiple convolutional filters, and each filter is scanning over the sequence and at each position looking for a specific sequence pattern And these sequence patterns are transcription factor binding preferences. And in this toy example, we have only one convolutional layer, but usually we use models with multiple convolutional layers, and this allows us to learn combinations of transcription factor binding sites that tend to be associated either with enhancer activity in our tissue of interest or a lack of enhancer activity in that tissue. After that, we have what's called a max pooling layer. And this encodes the assumption that within a short region, we expect to see a transcription factor binding site only once. And what this does is it takes all the outputs from each position in a short region and then just outputs the maximum output. And this also has a nice advantage that for other parts in the network, we then have fewer inputs, which requires our model to not have to learn as many parameters, which allows us to train our model on less data. So after that, we have what's called the fully connected layer, which then learns linear combinations of the outputs from our convolutional layer and how important each of those outputs are. And we often use actually multiple fully connected layers, which allows us to learn more combinatorial combinations of these different features and how important these different combinations are. Finally, we have our enhancer activity prediction, which, as I mentioned, is a number in the 0 to 1 range, which is the probability that our sequence is an enhancer. So we chose to use convolutional neural networks because they have many advantages. First, they do not require hand-engineered sequence features because we're giving our models, as I mentioned, a matrix that represents our raw DNA sequence. And this is a huge advantage because for the brain, and this is actually true for basically every tissue and cell type right now, we don't actually know what all of the sequence patterns are that are associated with enhancer activity. Second, as I mentioned, because of having multiple layers, we are able to learn nonlinear combinations of sequence features, which we know is important for brain enhancer activity. Finally, multiple papers have come out over the last few years showing the success of convolutional neural networks for related tasks, and here you can see a few examples. So now I've explained to you why we chose to use convolutional neural networks to learn what separates sequence patterns in enhancers from sequence patterns in non-enhancers. There are still some more important parts of the model setup that we needed to decide. First, we needed to decide how to define our positive set. How do we actually define an enhancer sequence? And we chose to take the ATAC peaks, and specifically we took the peak summit, so that is the location within the peak that has the maximum ATAC signal, and we went 250 base pairs in each direction. Our choice of 500 base pairs was not actually random, but we actually put a lot of thought into choosing how long we wanted our sequences to be. And we've had a lot of reasons for settling at 500 base pairs. First, ATAC peaks actually tend to be approximately 500 base pairs. Shorter sequences are less likely to go up the ends of scaffolds and highly fragmented genome assemblies. And Zoonomia was really part of why Zoonomia was able to get so many species is that many of our genomes are actually fairly low coverage and fairly fragmented, which allowed us to take advantage of DNA samples where we cannot actually get it from species, we cannot actually get a large amount of DNA. In addition, shorter sequences are more likely to fully map to distantly related species, and especially to fully map to species with fragmented genome assemblies. Finally, we can experimentally validate predictions for 500 base pair sequences in reporter assays, which is something that is not, with the current technology, feasible for sequences much longer than 500 base pairs. And I'm going to tell you a little more about how we're doing this later in my talk. But that still left the question, what negative set should we be using? And there are a number of negative sets that have been used for related tasks in the literature. Some people use flanking regions of enhancers. Others use random regions of the genome that are not enhancers in the tissue of interest, but have the same GC and repeat match contents. And there's different controversies about just how many we should be using in the negative set. Some use enhancers in other tissues that are not for enhancers, and some use the nucleotide shuffled regions. So these are shuffles of the enhancer sequences where the frequency of pairs of nucleotides is preserved. And we actually tried all of these negative sets, but we decided to actually use a different one that we developed that we found worked even better. And this was a negative set that was designed to specifically do what we wanted to do. And that is to predict whether a difference in DNA sequence between two species is going to be lead to a difference in enhancer activity. So this negative set is what we call non-enhancer orthologs of enhancers. These are regions of the genome that are not brain enhancers, but the ortholog, the corresponding region in another species for which we have data, is a brain enhancer. And we obtained these negatives using a combination of a publicly available tool called Leftover and a tool that our lab developed called Helper with the Zoonomia alignment. And Helper was a tool whose development I led in collaboration with Aaron Zhang, a former undergrad in the lab, and Morgan Worthland, a former postdoc in the lab, and you can check it out on GitHub. So now I'm going to tell you first about our machine learning models trained using only sequences from the mass. And you might be wondering, why did we start with training a model using data from only one species when, as I mentioned, we had data from 4 different species? And the reason for doing this is that we needed to show that we could train a model on data from one species and get good performance in another species by comparing our results to the actual data we have. Because ultimately, we're going to be making predictions in species where we don't have data. And if we haven't shown that we can get good performance in a species where the data wasn't used in model training, then we're not going to actually know if we can trust our predictions in a new species. And our class imbalance for this problem was 1.2 negatives for every 1 positive. So I'm now going to take a short deviation from the TACIT methodology to explain how we evaluate machine learning models with TACIT. And actually coming up with the right evaluation criteria actually took a fair amount of work because we were using these machine learning models to answer a new question. And therefore need to make sure that we were evaluating our models in a way that actually enabled us to figure out whether we could address the question we set out to ask. For the purposes of this talk, I'm going to illustrate my results as violin plots, which are smooth histograms. And just something I want to point out is that this dark orange line in this toy violin plot is the range of the data. And sometimes due to the smoothing, it can look like the data is outside the range when it isn't. So all of our predictions and all of our fast-con scores are in the 0 to 1 range, but the smoothing sometimes makes it look like things are a little bit outside of that range. So what I'm going to show you for most of the talk are machine learning models trained using only mouse sequences and the evaluations we did using sequences from rhesus macaque, where no sequence from rhesus macaque was used in model training. And this really shows the ability of a model trained in one species to make a prediction in a species whose sequences it has never seen. So the first evaluation I show you is the set that I call mouse plus macaque. These are brain enhancers in macaque whose orthologs in mouse are also brain enhancers. And this allows us to evaluate whether we can actually predict that enhancer activity is conserved. So the first thing I want to do is look at the fast-con scores of these enhancers. Now, I previously showed you an example of an enhancer whose activity is conserved, but the fast-con score is low. And I wanted to know, is this actually common? And what we would expect if the fast-con scores are actually indicative of brain enhancer activity conservation is that they would be close to 1. That's the highest they can be, and the enhancer activity is conserved. But you look, it's actually a general trend. Most of these fast-con scores are close to 0, showing that the nucleotide level sequence conservation is actually a poor indicator of enhancer activity conservation. We also used another method called Phylop. Phylop scores are negative log p-values. So a high positive value means high conservation, negative value means low conservation. We expected, since enhancer activity is conserved, if Phylop scores were useful, then we usually see very large Phylop values. What you see here is that they're actually only a little larger than 0, showing that Phylop isn't actually that helpful most of the time for determining whether brain enhancer activity is conserved. Fortunately, our convolutional neural network models tend to make large predictions. So this plus at the top indicates you would expect positive predictions. And you can see that most of our predictions here are pretty close to 1. We actually had an accuracy of 72%, showing that our convolutional neural networks can often overcome the limitation of conservation scores and actually tell us what brain enhancer activity is conserved. However, we didn't just want to evaluate our models on regions of the genome with enhancer activity conservation. Because at the end of the day, it's the regions of the genome whose enhancer activity is not always conserved that are likely to be causing differences in phenotypes. So to evaluate this, we focus on two different groups of enhancer groups. First, we look at what we call species-specific enhancers. These are enhancers in one species where the ortholog in another closely related species for which we have data is not enhancer. We also looked at clade-specific enhancers. And these are enhancers where all of the orthologs and the clade for which we have data are enhancers, but the orthologs in other clades are not enhancers. And for the purposes of this talk, I can tell you about our results for clade-specific enhancers. I'd be happy to tell you our results for species-specific enhancers offline. And what I want to point out is these were totally new evaluations that we developed. The previous work just looked at overall performance separately in each species without separating these into these three groups. And if one of these groups was actually most of the enhancers, the results might actually look great, even if the models were doing really badly on another one of the groups. And once again, for these evaluations, I'm going to show you machine learning models trained using only mouse sequencing and look at how they do on macaque sequences where no macaque DNA sequences were used in training. So the first step I'm going to describe is what I call macaque not equal to mouse. These are macaque enhancers whose mouse orthologs are not enhancers. And we're really pleased to see that we generally got positive predictions here. We achieved an accuracy of 77%. And this is really amazing because these are macaque enhancers where the mouse ortholog is not an enhancer. So it's not, the model saw only mouse sequences in training and yet was able to predict that the macaque ortholog, whose enhancer activity differs from mouse ortholog, actually is an enhancer. So we then look at the set that I call mouse not equal to macaque. And these are non-brain enhancers in macaque whose mouse orthologs are enhancers. And here is indicated by the minus on the top of the violin plot. We would expect small predictions because we're validating on macaque sequences that are not enhancers. And we were really pleased to see that our predictions were generally low. We achieved for this task an accuracy of 86%. And I should note that our conservation scores here can't really differentiate between these two groups because conservation scores tell us how conserved at the nucleotide level is a region. They don't tell you in which species is the region's function conserved. So in addition to evaluating our results on clade and species-specific enhancers, we also want to evaluate our results on tissue-specific enhancers to show that we could overcome conservation scores limitation of tissue agnosticity. In other words, if we look at these liver enhancers, we should be predicting that they are not brain enhancers because our model is trained to predict brain enhancer activity and not enhancer activity in general. So to do this, we once again trained machine learning models on mouse and evaluated on macaque sequences where only mouse sequences were used in training. And the additional validation that we did is these models were trained on the brain. We evaluated performance on the liver, a tissue not used in training, to see if we could figure out which of the liver enhancers are brain enhancers. So the first set I'm going to tell you about is what I call brain not equal to liver. These are brain enhancers that are not liver enhancers. And this allows us to evaluate whether our model has learned sequence signatures that are specific to brain enhancers and not shared in every tissue. And we're really pleased to see that our model generally got large predictions for this task, achieving an accuracy of 80%. We then evaluated on a set that I call brain plus liver. These are enhancers that are regions that are enhancers in both the brain and the liver. And this allows us to evaluate if our model can also learn sequence signatures that are shared across enhancers in multiple tissues. And we're really pleased to see that once again, our model predicted most largely predictions close to 1, achieving an accuracy of 88%. Finally, that is the set that I call liver not equal to brain. These are liver enhancers that are not brain enhancers. And we would expect here is to get small predictions because these are not brain enhancers and our model is trained to learn brain enhancer sequence signatures and not sequence signatures of any enhancer regardless of tissue. And we were extremely pleased to see that we do generally get small predictions, achieving 91% accuracy on this task. What you can see here is that conservation scores are actually not that different between these three groups, showing that, as I mentioned before, conservation scores don't tell you about tissue-specific function conservation. Finally, we wanted to see if it was really worthwhile to generate data from multiple species. So the way that we did this is we put, instead of just training models on mouse sequences, we used our data from all of our species, training models and data and piece of brain for 4 species. And we found that the models trained using data from all of the species do a little bit better. However, this is not actually an ideal evaluation, because what we really want to see is if we train a model on one species using data from one species versus using data from another, can we get performance on yet another species not using training? So the way that we evaluated this was using our liver data. So the data our lab generated came from mouse, macaque, and rat. So what we did is we trained 2 different models. First, a model using only sequences from mouse, and then a model using data from all 3 species. And then we used publicly available data from cow and pig from the FANG Consortium to evaluate these models, where our positives were enhancers in cow and pig, whose orthologs are not enhancers in any of our species. So this is a clade-specific enhancer for Boreoeutherian. And our negatives were not enhancers in cow and pig, whose orthologs are enhancers in all of our species. And here we're evaluating performance using 2 metrics that use a lot of machine learning called AUC and AUPRC. The values of these can be between 0 and 1. At random for AUC, you'd expect a performance of 0.5, and at random for AUPRC, you'd expect a performance of the size of your minority class, which is, in this case, was less than 0.5. So you can see that both our models are doing a lot better than random, but by training on data from 3 species, we do get a nice boost in performance, showing that it really was valuable to get data from a few more species. Finally, we were very happy to see that our models did correctly make predictions about the macaque orthologs of the 2 enhancers I showed at the beginning. So you can see that for this enhancer, whose ortholog is a macaque enhancer, we do get a prediction of 0.92, very close to 1. This enhancer with the high conservation scores, whose macaque ortholog is not enhancer, we get a very small prediction of 0.02. You can learn more about this work and some other evaluation criteria I didn't have time to go into in our paper that recently came out in BMC Genomics. Finally, I'll show you the performance for all the metrics I described for our multi-species motor cortex enhancer model. So as I mentioned for AUC, we'd expect a random performance of 0.5 and a best performance of 1. You can see we're doing well on all of our metrics. For UPRC, the white bars here indicate we'd expect at random. The best possible performance is 1. You can see once again, we're doing really well. So now that I've told you about how we evaluate our machine learning models, I'm going to turn to telling you some more details about how we implemented the rest of TACIT for brain size. And I'm going to specifically go on to our 3rd step of TACIT. So to identify orthologs of enhancers, we used Zoonomia's new alignment, and we chose 222 Boreoeutherian mammals. The Boreoeutheria are the mammals in the black boxes, and the reason we limited to Boreoeutheria is that we don't actually have any enhancer activity data outside of Boreoeutheria, so we can't actually evaluate whether machine learning models work in additional species. And this Zoonomia alignment is called a Cactus alignment. And the reason we had a number of reasons for wanting to identify orthologs using a Cactus alignment. First, the Cactus alignment is reference-free. And by not having a reference species, previous alignments said, let's just keep the human genome and align all of our other species to that. By not having a reference, we can actually trace orthologs of enhancers that are completely lost in the common species used for references, which are human and house mouse, such as bat enhancers that have no orthologs in human, or that human-specific deletion I mentioned before. In addition, the Cactus alignment method can account for structural rearrangements such as inversions that are not accounted for by previous alignment methods. And we actually have found at least one example of an enhancer that is inverted, so it's in different orientations in different species. And we would never have been able to trace that enhancer across multiple species using an earlier alignment. And once again, we obtained our orthologs using Howlett, Dover, and Halpern. So now that we have predictions in about 100,000 motor cortex enhancers as orthologs in 222 mammals, we need to find a way to associate these predictions with phenotypes. And for that, I'm going to describe the last 2 steps of TACIT. This was work that I did in collaboration with Daniel Schaefer. Daniel is an amazing undergraduate who's planning to apply to PhD programs next fall. And if any of you are looking to recruit PhD students, I want to tell you, you'd be super lucky to get them in your lab. So we have, to explain what motivated the way we chose to do this, I want us to consider this toy example of a macaque enhancer. You can see its predictions at orthologs in a bunch of different species. And now let's consider this toy example of a phenotype. Now what you can see is that there's a really strong positive association between our predictions and our phenotype. Now at first glance, this looks like a super exciting result that should be a top priority for further investigation. However, if we look at it a little more closely, what we can also see is that there's a really strong negative correlation between our predictions and how closely a species is related to rhesus macaque. Thus, it is impossible to know if this enhancer is actually involved in our phenotype or if this result is confounded by the phylogeny. So we therefore decided for our associations to use a method called phylogeny, which is a sped-up version of phylogenetic generalized least squares. And the idea is we use the generalized least squares regression to associate our predictions with a phenotype, but we have an expected variance covariance matrix that is based on phylogenetic relationships between species. And for binary phenotypes, we used a logistic version of phylogeny called Phylogeny. We then needed to find a way to quantify the significance of our associations. And to do this, we used a special kind of permutation test called phylogenetic permutations. And the way that this works is we create a null distribution by permuting our phenotypes. And we do these permutations in a way that preserves the higher level phenotype tree topology and therefore gives us a more realistic empirical null. So Daniel tried to do this, and what he found is that this didn't really scale up well to over 100,000 in motor cortex enhancers for binary phenotypes. So what he did is he implemented a speedup that for binary traits was able to decrease the runtime by over an order of magnitude. And if you're interested, you can check this speedup out on GitHub. So now that I've described how we implemented TACIT for brain size, I'm going to tell you about some of our results. We identified 34 motor cortex enhancers associated with the evolution of brain size residual. So Daniel did a massive literature review on the genes near these 34 enhancers. And he found that 8 of them are near genes associated with micro or macrocephaly, which is having especially large or small brains in humans. So these genes are actually, if you see a mutation in human, that mutation seems to cause having a very large or very small head. So in fact, he found that enhancers near micro or macrocephaly associated genes tend to have stronger associations with brain size than other enhancers. He also found that 18 enhancers are near genes associated with neurodevelopmental disorders. And he found that 5 are near genes implicated in the growth or spread of brain cancer. So here you can see an example of one of these enhancers. On the x-axis, we have our predicted motor cortex enhancer activity. On the y-axis, we have our brain size residual. And what I plotted are just, to make this plot cleaner, just the points for cetaceans. And you can see here, we have a really strong negative association. And as I mentioned before, cetaceans have the largest brain variation in brain size residual of any mammalian order, and we previously had no idea what the mechanism was underlying this variation. So to zoom in on this enhancer a little more, here we have our predictions in our cetaceans and our brain size residual. So we have here the bottlenose dolphin and its closest relative, the killer whale. We can see a large difference in brain size residual, and that is associated with a large difference in the opposite direction in our predictions, which is what we'd expect because this is a negative relationship. We can also look at the harbor porpoise and its closest relative, and once again, we see a difference in prediction, a large difference in the opposite direction. Finally, what we can do is look at 2 marine mammals that both have large negative brain size residuals. These are 2 mink whales, but one is much more negative than the other. And once again, we see a prediction difference, while small, that is in the opposite direction. So the question is, well, where is this brain size enhancer? So here, this is a browser shot of our mink whale. You can see the common mink whale. Here in this red box is our enhancer, and it is near the gene LRIG1. So we then looked at the gene LRIG1 in some mouse 3D interaction data. We've heard some talks on AC and how this allows us to figure out what enhancers are interacting with which transcription start sites. So we looked in mouse because mice have small brain size residuals and the relationship with brain size residual is negative. And Daniel found that there is a statistically significant physical interaction. And this was actually a really exciting result because our mouse data was really shallow and there weren't very many statistically significant interactions. Yet one of the few interactions was this enhancer where the transcription starts at LRID1. LRID1 is expressed in the mouse cerebral cortex, and LRID1 regulates quiescence in adult neurostem cells and neurostem cell proliferation during development. Another example is this enhancer, which is negatively associated with brain size residual in the great apes. And this was not the enhancer mentioned in the previous study because it does have an ortholog in human. So here's a browser shot of the chimpanzee. You can see this enhancer, and this enhancer is near the genes MED17 and VSTM5. So MED17, VSTM5, and CAP1D are all expressed in the mouse cerebral cortex, and as I mentioned, mice have small brains. You want to look at mouse because this is a negative association. Mutations in MED17 are associated with microcephaly and other neurodevelopmental disorders, and VSTM5 regulates neuronal morphology and vibration. So we then wanted to know, why is it that the human ortholog is very confidently predicted to be a non-enhancer, but the ortholog in chimpanzee, our closest relative, is confidently predicted to be an enhancer? So as pointed out last talk, you can actually interpret machine learning models to figure out what the important features are. So we did this and looked at the important sequence features. And then we looked for these sequence features in this particular enhancer, and this is what we found. Now this sequence actually matches the binding preference for the transcription factor CTCF. And as was mentioned in the previous talk, CTCF plays an extremely important role in connecting enhancers to transcription start sites. So CTCF is really important for enhancer activity in the brain, and in fact in almost every tissue and cell type. Now, if we look at the human ortholog of this specific sequence, what we see is that this G is an A. And if you look at the CTCF binding sequence, you can see in some positions there are actually multiple options for what sequence you can have, but in this particular position, G is the only option. So what that suggests is it's possible this human ortholog is not an enhancer because it cannot be bound by CTCF. Now that I've told you about some of our results for applying TACIT to brain size, I'm going to tell you about some of our findings for applying TACIT to other phenotypes. So we have applied TACIT to a number of other phenotypes. We currently have a number in review. In addition to brain size, we have vocal learning, solitary living, group living, and sleep patterns. And we also have been discussing trying to extend TACIT to additional phenotypes, including longevity, and also working with the Hindle lab at UNLV to see if we can apply this to hibernation. And what I should mention is that for all of these phenotypes, in order to use TACIT, other scientists did a huge amount of work curating phenotype annotations that were essential for this work. And also, if you know of other phenotypes with their annotations, and there's open chromatin data publicly available, please get in touch. I'd be interested in collaborating. So I can't tell you about all of our results today, but I'm going to focus specifically on vocal learning, where we have some really exciting results. Vocal learning is a really interesting phenotype for us to study with TACIT because it has evolved convergently at multiple levels. So at the behavioral level, we have vocal learning abilities, as do songbirds and parrots and hummingbirds, but our closest relative, the chimpanzee, does not have vocal learning abilities. Neither do doves, which are much more closely related to these birds. At the neuroanatomical level, a previous study found that the neural circuitry involved in vocal learning in humans is actually the same as the neural circuitry involved in vocal learning in songbirds. In addition, a previous study looked at gene expression associated with the evolution of vocal learning and found a set of genes in a few different brain regions whose expression is associated with whether a species is a vocal learner, and one of those brain regions was the motor cortex. So to enable us to do this, my lab mate Morgan Worthland curated phenotype annotations for 181 Boreoeutherians in the Zoonomia dataset in terms of whether they're vocal learners. And this then allows us to ask, how has this gene expression convergently evolved in vocal learners? So what are the enhancers regulating the genes found in the study? So we applied TACIT for motor cortex to vocal learning and got a few dozen enhancers associated with the evolution of vocal learning. I'm only going to have time today to tell you about a couple. Happy to tell you about more offline. But here is one example. So this is a binary phenotype. So we have here is a histogram, a smooth histogram of our predictions for vocal learners in the magenta, and a smooth histogram of our predictions for vocal non-learners in the gray. You can see, and then in dots here are all of our vocal learning clades. You can see that all of our vocal learners are, or the orthologs from our vocal learners are predicted to be non-enhancers, and that there's a huge shift towards the left in terms of predictions for vocal learners relative to predictions for vocal non-learners. So this enhancer is near the gene VIP, and VIP is actually downregulated in the hummingbird RA, which is the bird brain region corresponding to male motor cortex, where hummingbirds are vocal learners, but it's not downregulated in the dove RA, where doves are not vocal learners. In addition, VIP expressing neurons in mouse motor cortex send projections to the striatum, and corticostriatal projections are involved in vocal learning and production. Another example is this enhancer, negatively associated with vocal learning, and this is near the genes TSHC3 and ZNF536. Now, Morgan did also for vocal learning, like she did for brain size, protein coding RAR converge. But unlike what happened for brain size, where she found no results, she actually did find a few genes with relative evolutionary rates associated with the evolution of vocal learning. And two of her top hits were TSHC3 and ZNF536. So here on the x-axis, we have a relative evolutionary rate. Each row here is a species. In magenta, we have our vocal learners. In our gray, we have our non-vocal learners. You see that our vocal learners tend to have high relative evolutionary rates in comparison to our non-vocal learners. So what this suggests is that we have these two genes that are accelerating evolution, so potentially becoming less functional in vocal learners. And in concert, there is an enhancer near these genes that is losing activity in vocal learners. And what that shows us is, as I mentioned before, we have two ways of identifying sequences associated with evolution of phenotypic diversity. One by comparing protein coding sequences, and another by comparing enhancer sequences. But by using these methods together and comparing the results, we can gain even more detailed understandings into the mechanisms through which phenotypes have evolved. So Morgan did some literature search into these genes, and she found that human orthologs of TSHC3 and ZNF536 are in a locus associated with severe species stability, and the disruption of TSHC3 in humans impacts corticostriatal circuits. Finally, we are starting to experimentally validate some of our predictions in a massively parallel reporter assay. So this is experimental work that has been led by our amazing lab manager, Ashley Brown, and it's a brand new technology. And the idea is that what we do is we synthesize our enhancer orthologs, where for some we predict that they're enhancers and for some we predict that they're not. Then what we do is we put them into plasmids, these are circles of DNA, where we have our enhancer ortholog fused to a minimal promoter fused to a random sequence, which we call a barcode. And then, so previous studies have then delivered these into cells in a dish. And what Ashley has done is found a way to deliver these into a mouse where it spreads throughout the mouse, including to the brain. And this is by putting them into a virus that can cross the blood-brain barrier. And by doing this, we can then inject these sequences into the mouse without actually having to drill any holes in the brain, so without doing any kind of disruption to the brain. So this is super exciting. And then what it does is it allows us to compare results in different regions within the brain of the mouse, as well as in other tissues, without being confounded by which mouse we're using. So what we can do is we can then sequence the RNA from these plasmids. And if an enhancer is capable of activating gene expression, we'd expect to see a lot of RNA from the barcode. But if the enhancer is not capable of activating gene expression, we should see hardly any RNA from the barcode. So then what we can do is we can compare the amount of RNA from the different barcodes to see if our predictions are right in terms of which regions are actually enhancers. And we're now starting to do this for some of our orthologs of our enhancers associated with both vocal learning and brain size. Finally, I previously mentioned that it's very hard to study clade-specific enhancers using TACIT because if an enhancer is predicted active in just one clade, we can't figure out if associations are real or if they're just confounded by the phylogeny. But we can actually do some investigation into clade-specific enhancers. So the way that we do this, this is work that I came up with that was implemented by Daniel, is we take our enhancers and we have as features our predictions, and then we cluster them. So each cluster of enhancers, like the one seen here, is a group of enhancers that's predicted to be active in either one or more clades. And then what we can do is we can take these clusters and overlap them with enhancers whose functions we have already investigated. So what we did is we took our clusters and overlapped them with mouse enhancers that have been associated in a previous study with neuron firing. When we found that these enhancers associated with neuron firing tended to overlap the cluster with predicted mRNA-specific activity. We also did this for human enhancers associated with neuron activity. And we found that those enhancers tended to overlap a cluster that is with enhancers supposed to be active in just primates and carnivora. So what this suggests is that enhancers associated with neuron firing may have clade-specific activity. In conclusion, we have developed TACIT, which associates predicted enhancer activity of candidate enhancers with orthologs with phenotypes. We use TACIT to identify 34 motor cortex enhancers associated with the evolution of brain size residual, and additional enhancers associated with the evolution of other neurological phenotypes. TACIT can be applied to any phenotype with annotations in dozens of species and with enhancer activity data in a relevant tissue or cell type available from at least a few species. With that, I'd like to thank my postdoc advisor, Andreas Fenning, my many lab mates who contributed to this project, especially Daniel Schaefer, Alyssa Waller, Morgan Worthley, and Ashley Brown, our funding and compute resources, and our many amazing collaborators in the Zoonomia Consortium. I'd be happy to take any questions. Yes.
I'm curious if during the training of this model, when you incorporated data from other species, you identified species-specific sequence features that impact the predictions of the model. And the reason I'm going to ask is because for predicting 3D structure, when we first applied the models from mouse, they didn't have comparable. I thought it was because mice have a species-specific expansion of B2 signs. So I'm curious if
Elaine, that is a great question. So I actually did interpret the model for the mouse-only model and the model for data for multiple species. I did find more sequence features in the model for multiple species. When I did a lit review, those sequence features are actually also important for brain enhancer activity in the mouse. And what it seems to be is that those sequence features occur less frequently. So we just didn't have enough training examples in the mouse-only model for the model to pick it up. But it wasn't actually learning sequence features of species-specific enhancer activity. And what I can tell you is I showed you situations where we actually, based on our models in both performance and interpretation, we think there is a very highly conserved regulatory code. And for things that aren't conserved, there are actually enough examples for our model to pick it up. We do have some situations where we train a model on one species, and the performance on a species not used in training is poor. And yet when we put all the data together, we get amazing performance. We are currently in the process of interpreting those models. So we think those models that we actually have different regulatory codes in different species. The model from data from multiple species is probably learning thing again. Any other questions? Feel free to come find me at lunch or later in the day or email me and ask me more questions.
Okay, so lunch is going to start pretty soon, and
Is that clear? Okay, fine.